Welcome back, Bio3D Prep, for another lecture on the digestive system. Here we're going to be looking at the anatomy of the digestive system and how it relates to absorption. In your textbook, it's page 226 to 231. By the end of this lecture, you'll be able to identify the role of the gallbladder and liver in digestion, identify where molecules enter the circulatory system from the digestive system, and describe how molecules are transported to the circulatory system from the digestive system. You'll also be able to explain how digestive activities are regulated by hormones. So the liver and gallbladder, these are two accessory organs. Along with the pancreas is also an accessory organ. The liver has two functions related to digestion. One is the production of bile salts. The other is it's a storehouse for glucagon and some, or glycogen I should say, glycogen and some vitamins. So this is really an after digestion, because it's already been absorbed. So after digestion and absorption. Whereas this is the actual aiding digestion. The liver can also convert poisons into usable materials and waste products that aren't as harmful as the original waste products. For example, our liver can convert ammonia, which is NH3, into urea. And this is really good. Ammonia is extremely poisonous. Something like 5 micrograms will kill you. Once it's converted into urea, once those ammonia molecules are stored into something bigger, a lot more of it can be stored in our blood before it has to be excreted through our kidneys and eventually through our bladder. The gallbladder stores the bile salts produced by our liver. So our liver produces bile salts, our gallbladder stores them, and in the presence of fats, when fats enter our small intestine, there's a hormone secretion. These hormones enter our blood, travel to our gallbladder, and the gallbladder releases the bile salts. The bile salts enter the small intestine, and they physically break apart fat globules. And I make this analogous to, so there's no chemical digestion. This is the important part. It's physical digestion. If you think about, I guess an analogy would be, for example, when you cook spaghetti, after you haul the spaghetti out of the boiling water, especially if you want it to sit for a while, you add olive oil to it or some kind of oil. And what that does is it keeps those long noodles from getting all twisted and stuck together with each other. Now, spaghetti is a carbohydrate, so it's a different chemical, but the analogy is the same. That bile salt allows all those fat chains to untangle from each other, but there's no chemical digestion yet. But what it does do is it spreads out those long fat chains and allows digestive enzymes to attack them more efficiently. So it's about making it more efficient fat digestion. Once we get to the small intestine, what we're really going to talk about is absorption. So here we have a picture of a capillary again. Inside each capillary is, a picture of a villi again. Inside each villi is a capillary and a lymph vessel. And notice the capillary is the red and the blue. And blood's going to come in through the artery. The oxygen is going to go out and give oxygen to all of our digestive cells. But nutrients are going to enter our circulatory system or our lymph system, depending on the nutrient we're talking about. So most absorption occurs through the walls of the small intestine, the walls of the walls. That ain't good, so we'll go like this. The walls of the small intestine. Through the walls of the small intestine. On your notes that will be gone, but in the lecture it will last forever. Here we go. These walls, they're covered with those finger-like projections called villi, increasing surface area by a factor of 10. Each villus is supplied with a blood capillary and a lymph vessel called a lacteal. The outer layer of cells are also folded into finger-like projections. Those are the microvilli we saw in the last lecture. Again, increasing surface area, and here is the purpose. All the carbohydrates and proteins that we broke down So all those carbs and proteins are absorbed into the capillaries, and really these are going to be single sugar units and amino acids, because all of those proteins have been broken down into amino acids. The fats are going to be absorbed into those lacteals or into our lymph. And they require special packaging because carbs and proteins are water soluble. They dissolve in water. Our blood and our stomach are full of our blood, and our small intestines, our body, are full of water. So carbs and proteins traveling through water, not a problem. They dissolve in the water and can be transported. Fats, however, we all know fats don't dissolve in water. If you think about when you put oil inside a pot of water, it floats on the top. Or you think about animal fat. If you boil, say you're boiling chicken bones to make your chicken soup, there gets that layer of fat floating on the top, a layer of oil, and then if you let it cool down, it forms a nice layer of fat on top of the broth. So fats don't dissolve in water, so they're going to require special packaging. When we look at the absorption of glucose, here's a summary of what's going to happen. We have our starch, our polysaccharide. Each one of these green circles represents a single sugar unit. We have some enzyme action. For example, amylase is going to break that starch into disaccharides, and those disaccharides by carbohydrates are going to be broken down into single sugar units, which can then be absorbed by our intestinal wall cells. Notice each cell has these little waves here. Where's my pen? I've lost it. Here we go. These are those microvilli. Each one is called a microvillus. And active transport, which means it takes a little bit of energy to get these things to go from our small intestine into our blood vessels. And it's active transport. These are energy molecules, right? Glucose is our energy source. We want every drop of glucose or every monosaccharide to go from our intestine into our blood. Here's the words that go along with the previous picture. Digestion of starches or complex carbohydrates. It begins in the mouth with salivary amylase. In the stomach, the pH changes at salivary amylase, making it stop working. The chyme from our stomach enters a duodenum. The pH rises to 8. And then the pancreatic amylase is going to complete the digestion of starch to disaccharides. Those disaccharides are going to be attacked by carbohydrates. Those carbohydrates are going to hydrolyze disaccharides, turning them into monosaccharides such as glucose or fructose or maybe ribose, depending on the size of the sugar unit. Those monosaccharides are absorbed by active transport into our intestinal villi, or from our intestinal villi into our circulatory system. They enter the bloodstream. They're transported directly to our liver, and our liver is going to convert all the monosaccharides that aren't glucose into glucose, because glucose is a version of sugar that we use to get things done. So if we're eating something that's no longer not a glucose shape, it's going to be converted to a glucose shape, and then it can go out to all of our body cells. That's our source of energy. And now we're going to look at amino acids. Now amino acids, same kind of picture. We have a protein, a great big long chain of amino acids. This chain is going to be attacked by things like trypsin. And this protein here would be a peptone. It's already left the stomach, so it's shorter than it used to be, right? It's been attacked by pepsin. It's become a peptone. It gets attacked by trypsin or chymotrypsin. These things break it down into small peptide chains, and then we have our peptidases. Our peptidases break down those peptides into amino acids, and the amino acids are absorbed into our blood vessels by active transport. And again, here's the words for the picture. Digestion of proteins. The polypeptides that are produced by the action of trypsin in the stomach are further digested, chemical digestion, in the small intestine by proteases such as trypsin. or chymotrypsin. And these are secreted by the pancreas. The proteases are secreted as inactive enzymes and activated by different enzymes secreted into the small intestine. Both trypsin and chymotrypsin break down peptide bonds between specific but different amino acids. And so that's why we have, not only do we need a specific enzyme for each type of macromolecule, we need a specific enzyme for different bond types in the macromolecules. And then we have a whole bunch of peptidases splitting up those short peptide chains into single amino acids. The amino acids, just like the glucose, are absorbed by active transport into the villi of the small intestine and then transported from our blood into the liver. And then the liver can distribute those proteins out to the rest of our cells. Now, fat absorption is a little bit different. We still have our small intestine, and notice that we have fat as a globule of molecules. So each one of these molecules is a triglyceride. Here's one triglyceride. Bile is secreted from our gallbladder. And when the two mix, we get emulsification. And what that does is it just separates each of our triglycerides from each other, allowing lipases to break them down into glycerol and fatty acids. So, a couple things. This step here is physical digestion. The second step is our chemical digestion. They're absorbed into our small intestine and then brought into our lymph by diffusion. So there's the other big difference. Proteins and sugars are absorbed by active transport. Fats are absorbed by diffusion. So for lipids, digestion of fats, they arrive in the duodenum pretty much untouched in terms of chemical digestion. Bile is secreted, which emulsifies those fat droplets. It's a physical process. The covalent bonds that join glycerol and fatty acids are not broken. Hydrolysis is carried out by lipases that are secreted by the duodenum. The resulting glycerol and fatty acids are then absorbed into the cells of our villi by diffusion. In these cells, these fat subunits are reassembled into triglycerides, and then they're coated with a protein because fats are not soluble by themselves. So we have to make them soluble first in water. Then they can be transported in our lymph system. So that's the other big difference. They don't enter our circulatory system right away. They enter our lymph system. They're carried actually up to our chest, where they're dumped into the venous portion of our blood or into some veins. The protein coats are removed by lipases, and then the triglycerides are rehydrolyzed into their fatty acid and glycerol components, where they're then available to be used by body cells. Now, all of this has to be regulated by something. We don't just have enzymes floating around in our intestine and stomach waiting for something to happen. That wouldn't be efficient or useful. So it's all regulated by chemicals. So a couple of things that regulate when digestive enzymes are going to be secreted into our stomach or small intestine are, first of all, the presence of food. When we smell food, when we taste food, those signals go to our brain, and our brain tells our digestive system to start getting enzymes ready. The other thing that happens is there's some responses when food enters our stomach, for example. Our stomach sends out hormones that stimulate either our small intestine or our pancreas to produce enzymes or substances involved in digestion. So the presence of food plays a big role in when we secrete certain things. So for example, the presence of a bolus, as soon as we get a bolus of food even traveling down our esophagus, this stimulates the production of gastrin, which is a hormone. And gastrin then stimulates glands in the stomach lining to produce acid and pepsinogen. So gastrin's produced first, it's produced by our stomach, and then it triggers the other cells in our stomach to produce substances like the acid and pepsinogen. When chyme passes, from the stomach to the small intestine, a whole horde of hormones are released. Secretin, CCK, and GIP are all hormones that get dumped from our small intestine into our circulatory system, and then they travel throughout our entire body, but they're going to reach things like our stomach, our pancreas, and our gallbladder. So when these hormones reach our stomach, it actually slows our stomach down. It says, wait a minute, don't dump all this food directly into your small intestine yet. There isn't room for it. Keep it in the stomach. Let it do some more physical digestion before it enters the small intestine. Our pancreas is going to be stimulated because our pancreas releases all those digestive enzymes that break down, especially the final stages of chemical digestion. The other thing that's going to be produced by our small intestine are bicarbonate ions. These are really important. They're a base. And from Unit 1, we know bases neutralize acids, and that's important because this acid is coming from stomach, or from our stomach. If we don't neutralize it, a couple things are going to happen. The acid itself will damage our small intestine, but that chyme is full of pepsin, and pepsin is a protein digesting enzyme. It will damage our small intestine cells. Those villi cells will become damaged by that pepsin. That pepsin will start breaking it down, which is no good. We all know that the cell membrane, if you remember from our Unit 1, every cell membrane has those proteins that are integrated in the cell membrane for transport. Those are transport proteins, carrier proteins, channel proteins, glycoproteins, all those proteins with different jobs in the cell membrane. If we dump a bunch of protein digesting enzymes in the same place, well, those cells are going to get damaged. The other thing that's going to happen is the gallbladder will be stimulated to release bile. So we'll look at each of these three hormones a little more closely. All three are present in the duodenum, and they all decrease activity of the stomach, especially when chyme is entering and it's full of fat. So if it's entering the small intestine, and especially when it's fatty food, it's going to slow down our stomach processes, increasing time for digestion. Now secretin has another role. It's the one that's going to react to the acid. So we have this acid coming from our stomach into our small intestine. We want to make the pancreas release bicarbonate ions. It also deactivates the pepsin. Cholecystokinin, or CCK, and that's why they call it CCK, because cholecystokinin isn't always easy to say. It reacts in the presence of nutrients that in the chyme and increase just the release of digestive enzymes in general. So it's going to increase things like trypsin, chymotrypsin, proteases, carbohydrases, all those enzymes we need to speed up chemical digestion. These are all homeostatic mechanisms, so the response is going to neutralize the stimulus. That's the goal. When the stimulus is gone, then the response stops. Here's an example. If we take a look at this, and I've been talking about it for the last 3 or 4 slides now, but this acid and protein digesting enzymes coming from our stomach are a real problem for our small intestine. So the chyme mixture is going to damage our small intestine unless we neutralize that acid and destroy the pepsin. Here's the process. Bicarbonate ions from the pancreas enter our small intestine to protect it from that acid-soaked food in the stomach. The general process is prosecretin. So how does your small intestine know when acid is there? They don't talk. They don't have eyes. They don't have taste. So how do these little cells know what's going on? And so here's the answer. Inside your small intestine is a substance called prosecretin. And if you take a look, prosecretin, think about before being secretin, it's something else. As soon as the acid in your stomach hits it, it becomes secretin. So the acid turns it into secretin. And once that happens, it enters our circulatory system. So it becomes secretin, goes into our blood, and the blood carries it throughout our entire body. And part of our body is the pancreas. So it travels through all of our body, but the important part is it's going to reach our pancreas. Secretin reaches the pancreas. The pancreas gets this signal letting it know there's acid in our small intestine. So it releases bicarbonate ions, and these are that base that neutralize acids. The bicarbonate ions are produced, they go through the pancreatic duct directly into our small intestine. If we were to draw it as kind of a homeostatic chart, we get this chyme entering our small intestine. This acid converts prosecretin into secretin. The pancreas senses the presence of secretin, sends it out into, or the pancreas, sorry, we're kind of skipping a step here, but this is happening in small intestine. So, through our circulatory system, it reaches our pancreas. The pancreas then releases bicarbonate ions. Oh, look at that. That's what happens with a bad cut and paste. Holy moly. It's going to go through the pancreatic duct. And what it's going to do is response, body temperature rises. Bad cut and paste. Alright. What it's going to do is neutralize the acid. The acid levels decrease. No more conversion of prosecretin into secretin. Look at that. I fixed every box except for one. And there's our negative feedback. The negative feedback part is right here. Negative feedback. As that acid is neutralized, secretin stops being converted from prosecretin, and the whole chain stops once we've neutralized all the acid. Here's our accessory organs in kind of a schematic of how they interact with each other. Each of those three hormones is coded for with a color. And if you just take a look, for example, gastrin leaves our stomach, goes to our circulatory system, and then from our circulatory system just triggers other stomach cells to produce the acid in the pepsinogen. Secretin comes from the duodenum. Remember, it's prosecretin, it gets turned into secretin, goes into our small intestine. It does a couple things. The number one thing that we talked about is it triggers a pancreas to produce bicarbonate ions. Look at that. There we go. HCO3 negative. And then they get dumped. If you take a look here, this here is our pancreatic duct right here. It's going to dump those bicarbonate ions into our small intestine. It also triggers the secretin can also affect the gallbladder to produce bile. Didn't talk about that a lot. I really built on the other relationship instead. And CCK, same idea, comes from the duodenum. It triggers bile secretions. It also triggers enzyme secretions. When food enters our small intestine, these hormones go into our circulatory system and trigger our accessory organs to release digestive chemicals to break that food down. We finally made it to the colon. Chemical digestion is done. And most of absorption is done as well. We've absorbed all these nutrients into our body. So mostly our colon stores waste material, but there's also a bunch of water there as well. Every time we secrete an enzyme into our alimentary canal, there's water going in with it, right? Plus there's water in our saliva to moisten food. Water's produced to help get some of that chyme built up. So all that water gets reabsorbed back into our circulatory system so that we don't get dehydrated. There's also some bacteria. This is the part of you that's not you. That are living in our small large intestine. They help synthesize vitamins, specifically B and K from the waste material. Nervous system is going to monitor the waste levels. When you feel or when your bowels feel full, you will be stimulated to have a bowel movement and the waste is removed. Several factors influence the rate of digestion. We talked about sensory stimuli, sight, smell, and taste of food all cause these gastric secretions to start. Another example is when we have partially digested fats in the small intestine, enterogastrone, which is a hormone, actually slows down peristalsis. Fats are hard to break down, so our digestion slows down in order to give us more time to get nutrients from our fats. Hormonal control systems are throughout digestive system. We've talked about 3 in this lecture, and there are more. So, from this lecture, in terms of absorption, you should get the idea that glucose and amino acids, or are absorbed into capillaries found in our intestinal villi through active transport. We also got the idea that bile is secreted to emulsify the fats. It's physical digestion, and then those lipids enter our venous system through our lymphatic system. So it's actually two steps. First, they're absorbed into lymph by diffusion, no more active transport. Then they're packaged by proteins, and then they're carried to our circulatory system. And they don't actually enter our blood until they've been transported by lymph vessels up into our chest. There are a bunch of hormones, secretin, CCK, GIP, all are there to regulate digestive processes. They all decrease stomach activity to slow down the movement of food to increase digestion time. Secretin and CCK also stimulate the secretion of digestive enzymes. Bicarbonate ions, they are the thing that are going to save our small intestine. Coagulation of pepsin, neutralization of stomach acid, and it's produced by the pancreas and regulated by secretin. So secretin is related to the production of bicarbonate ions. Our large intestine, for the most part, is there for storing waste and reabsorbing water. And that's it. There's your digestive absorption.